All right. I think it's about time to get started. So let me welcome everybody to this last session of the second day of the CNI 2020 Fall Virtual Meeting. I'm Cliff Lynch, the director of CNI, and I just have a few quick introductory things to cover before turning it over to our speakers. We are recording this session, and this will be available after the session is complete through our usual channels. There is a chat box, and you are welcome to use that. Feel free to introduce yourself if you like, or to comment as the presentations proceed. There is a question and answer tool on the bottom of your screen, and please use that at any point during the presentation to queue up questions. Diane Goldenberg-Hart from CNI will be coming on after all the presentations and will moderate a question and answer session based on the questions that come in there. The only other thing I will mention is that normally these sessions do have closed captioning available, but there seems to be a glitch with this one. We should have closed captioning on the recording, and we may get it working here during the session. If we don't, my apologies. I think that's everything I need to say in terms of mechanics. At this point, I'd just like to welcome our four speakers, Nora Garza, Sarah Bowman, Jeremiah Trinidad-Christensen, and Florence Henderson. And I'm particularly delighted to have Florence back. Many of you will know her work from when she was at Internet 2. Some of you will also know her earlier work from when she was at IBM, but she is now the executive director of the Northeast Big Data Innovation Hub based at Columbia. And one of the things that I hope we'll cover at least a little bit today is the role of the big data innovation hubs, which I don't think is as widely known among the CNI community as it probably should be. This is an important strategic NSF initiative that actually touches on lots and lots of different projects. The primary focus, though, of this presentation is going to be the very rapid creation of the COVID Information Commons, which has been a sort of an amazing collaborative effort, like so many others that was born out of necessity as we've tried to respond to the challenges of the pandemic. And with that, I believe Florence is going to start off the presentation. So I will just thank our speakers one more time and hand it over to Florence.
Thank you very much, Cliff. And it's wonderful to be back with the CNI team. I've participated before, as you mentioned, and it's always a pleasure. So thank you very much for having us today. Today, we're going to be talking about the COVID Information Commons, and I'm joined by two of the COVID research principal investigators that have been awarded by NSF with COVID Rapid Grants, and Jeremiah, who leads our libraries team at the Columbia University. So the COVID Information Commons, it serves as an open resource to explore NSF-funded research addressing the COVID-19 pandemic. And that was particularly what we were asked to do when we received this COVID Rapid Grant. It is funded by the NSF Convergence Accelerator Program, which is one of the newer programs, which provides for multidisciplinary and interdisciplinary research across multiple of the domains that NSF funds. Next chart, please. So our agenda today, I'll give you a quick overview of the KIC, as we call it, the COVID Information Commons. Then Jeremiah will take us through the library's role. And we thought this was a great example of partnership between researchers, information technology, the big data hubs, and the libraries. Then two of our principal investigators that have spoken on our KIC community lightning talks are going to give their perspective and actually do their lightning talks. So you can see the range of research that's available through the COVID Info Commons and the community. Then I'm going to take you through a quick run through of the search and discovery mechanisms we have in the COVID Info Commons, and then we'll do our Q&A, which we'll all participate in. Next chart, please. So as Cliff mentioned, the Northeast Big Data Hub, which I'm the executive director for, is one of 4 big data hubs across the US that are funded by the National Science Foundation beginning in 2015. And then we got our next awards in 2019. So I lead the Northeast Big Data Innovation Hub at Columbia University, representing the Northeast US, the South Big Data Hub, the West Big Data Hub, and the Midwest Big Data Hub similarly are headquartered at a couple of universities and then serve their entire region. And this COVID Information Commons was a collaborative project across the 4 big data hubs. And that's part of what we're trying to do more and more is hold hands and work together to support our community around the challenges and opportunities in big data. Next chart, please. So a little bit of an overview. So the COVID Information Commons was created to increase accessibility to information, something we all care about in CNI. And this is specifically regarding the NSF COVID Rapid Research Awards, which are rapid response research grants. So when there's an emergency, they create these rapid awards. It could be for floods, fires, pandemics, things like that. And so that's what it was funded for initially. It now includes information for additional NSF grants that have COVID as a program name or a program reference code for those of you who know the innards of NSF parlance. And it facilitates knowledge sharing and collaboration across these COVID research efforts. It really started as, just give access to all this rapid information in one place. And it's evolved so much as a portal, as an international collaboration mechanism, which you'll hear from Sarah, as well as a community of collaboration, which you'll hear more about from Nora and from Sarah. So it's a resource for anybody around the planet, as you can tell by the international nature. And we have researchers, students, decision makers from academia, government, not-for-profits, everywhere really working together to leverage these research findings and to accelerate research to mitigate the societal impacts of the pandemic. We organize relevant information in multiple ways, if you could just go back one chart. And it allows us to look by research topic, institution, geography. And we recently, on October 30th, launched an upgrade that links to PI and research team data. Next chart, please. So this is a quick timeline. We were given the award, we were awarded in May of 2020, and we launched 2 search mechanisms, a simple search mechanism into the NSF database, as well as this cool machine learning generated map capability. Then in phase 2, which we launched between July and October, because we had the first launch in July, we delivered more search mechanisms. We have a Slack channel. We have a number of other deliverables. We have over 40 data sets that are actually shown and linked to for around the world, as well as these monthly COVID Info Commons webinars, KIC webinars. And we actually did this PI survey to get more data that we've included in the database, which is very valuable. And now we're looking at next steps, and we're going to talk more about that. Next chart, please. So COVID Info Commons is a portal and it's a community. So in the community, what we've done is when we first were going to do the launch webinar in July, we said, maybe we can ask a couple of PIs to present. And I actually begged one of them because I didn't know if anyone would say yes. Then before we knew it, 40 more people offered. So we said, oh my goodness, I guess they really want to do this. So in the launch webinar in July, we said, well, due to your enthusiasm, you are now part of the COVID Info Commons community, and we'll have monthly webinars until we get through all these lightning talks. And now we're scheduled at least through March. So we have hundreds of people in the KIC community, and you can sign up for it by going to covidinfocommons.net, which is our website. 73 members in our Slack channel and these monthly calls. And Nora graciously in our September call, when she presented, she said this, so we bottled it and we have it in all our presentations now. I'm sure she'll talk more about it, but she said, your site and the ability to come together is marvelous. I thank you especially for thinking about this and bringing us together. And people will be able to use your KIC site as a proper, safe, true information source. And that's really what we were hoping for. So as I say, as an Italian, I nearly cried when she said that, but we're so delighted that was the feeling. Next chart, please. And this is a totally open community. This is just quickly to see that, and you can have these charts, I put them into the Dropbox, that these are an example of some of the researchers that presented. Nora was in the first one. You can see the range here from UT Arlington to Princeton to UCSD, University of Southern California, all different directorates. Next chart, please. And then in October, Sarah, who's presenting today, presented. And it was interesting because during her talk, there was someone I saw asked a question. I'm like, oh, Sarah, Peter asked a question. She goes, I have like 3 others I'm answering right now. There was so much real-time collaboration. It was so exciting. Next chart, please. And then our next webinar is this Friday, or tomorrow, November 13th. And you're welcome to join that as well. Just go to covidinfocommons.net and register under events. Next chart, please. So since we launched it as a portal to find rapid grants, we also created a couple of other things. One is we created a tab called Meet the Researchers, really trying to humanize this. So we have human-to-human collaboration going on. And so what we've done is we've taken the separate lightning talks from each of the PIs that have done them, and we've put them in separately. We also connect them to their PI profile, which we'll talk about. Next chart, please. We've also started doing interviews with some of the researchers to ask them 5 questions about why are you doing this research? What have you found? What are you hoping to find? What collaboration opportunities do you have? Once again, humanizing it and creating the ability for people to find each other to enable each other's research and collaborate together. Next chart, please. So these are a few other resources on the KIC website, and you can go at any time. You could be playing with it in another screen right now if you want, covidinfocommons.net. So we have, under our resources, we have webinar videos, including a user tutorial for the Lingo4G Machine Learning Explorer map that I'll go through a little bit more. Then we have the July, September, October, and we'll add the videos as they come about. Next chart, please. We also have research funding opportunities around the globe, US and international. Next chart, please. We also have over 40 data sets from 6 continents. How cool is that? People send them to us because they know we have this portal. Next chart, please. We also have groups and guides, organizations and networks, guides and references. Next chart, please. And so all of that is provided by people who say, hey, include us, and we're delighted. So if you know of COVID-related resources, feel free to go to covidinfocommons.net, and we actually have a form that you can fill in to provide data. Under the events, we have prior events. You can see past events. We have the upcoming events. We have these monthly webinars because we have so many cool lightning talks lined up. Next chart, please. And this is our project team. So Jeanette Wing is our PI at Columbia University. She leads the Data Science Institute. I work in her team. And then my other 3 colleagues that lead the other big data hubs, Meredith Lee for the West Big Data Hub, John McMullen for the Midwest, and Renata Rawlings-Goss for the South Hub. Katie Nam is our operations manager. Helen Yang is one of our wonderful students. She's a junior at Columbia, and she's done a lot of the updates to the PI database. And then our Columbia University Libraries team and IT teams. And all together, we were able to pull this off, which is kind of incredible. And we're really delighted about it. And now we're looking at our next steps. Next chart, please. So now I'd like to hand it over to Jeremiah so he can talk about the library's role. And they were really important for us because they help us see some of the information science aspects that aren't as obvious to some of the rest of us. So Jeremiah.
All right, thank you, Florence. So this was a pretty good project to be working on. It was, at the time, working with something that did not fully exist yet. So a lot of the wards were in the same process as we were. So it meant a lot of room to kind of think and explore and try to understand what are the possibilities of what this could be. The libraries team that we have, they're listed here. All of us are within the digital scholarship unit within Columbia University Libraries. All of us have some level of expertise with institutional repositories, working with data, metadata, data management, even building web applications. The role that we were asked to do was to be able to come in, explore and think of ways for discovery for the information that was there. We did know from the beginning that we should be thinking very flexibly, that this is something that, although it was geared toward the rapid awards, there was room for growth in here. So in all of our thinking, we needed to keep that in mind, that it wasn't just for this information alone. So as part of that, we needed to understand what data was available from NSF. We need to also think through what are the additional information that might enhance in the data that's available through NSF that might be helpful. So we took a look at the existing data. The data came through both API and XML. We took a close look at what was available there. The XML actually had a lot more information there, but it wasn't updated as quickly. Whereas the information from the API was very regularly updated. So we compared, we assessed for the completeness, we tried to understand any errors or any problems that might be there, that might exist. We mapped out the various fields, their different relationships, thought through how each one might help somebody explore if they saw it readily in the front or in the back, or what are all those relationships. We also took a look at additional information that we might be able to pull in to enhance this. So we thought through, you know, if we brought in connected to information out of ORCID, what would that look like? Connect to other research that each individual researcher had already done or will do in the future. Making connections to Wikidata, for instance, for the institutions or other information that might be pulled out of there, out of like the abstracts, and be able to have that findable and showable. Same thing with geonames, whether or not we wanted to explore looking at building an interactive map that like lets people make selections, custom selections based off of whatever area they wanted to draw on the map. All of these kind of things we were looking at. Same thing with controlled vocabularies too. And then from the PIs, we knew that the survey was going to go out. So we needed to think through what's the information we wanted to get that might be really helpful. And the ORCID IDs were one of those to make sure that we connected the individuals that were listed there with who they are. Same thing with trying to understand any unstructured keywords that might happen in there. How do the researchers see their own work? How do they describe their own work? What are the keywords they apply to that research? And also the output too. Since this was so early on, there were no outputs at the time when we were looking at this. So really had to get an understanding of what we were looking at and how to display that through the site. So after we got roughly about 100 or so responses from the survey, the library team took a dive into the data and just focused very specifically on any of the websites that were named in there, as well as what type of output was going to be available. And we did a kind of cursory pass of this to try and kind of organize ourselves. So this is these couple charts are show you kind of what we were thinking at the moment. So with the URLs, we got URLs in there that some of these were associated with universities, some were .orgs, .coms, some were in GitHub. We pulled out the Google ones just because since these are things to exist, they did not exist at the moment. Some of the Google sites might be associated with login information from a particular institution or just from a few individuals. So we knew we might have to look at these later, but we needed to understand how many of these we had at that moment. We also wanted to look at the output itself, and there was such an amazing variety of output. Everything from data sets to things that would exist in social media, that might be a blog, that might be a video, that might be some kind of a conference or some kind of, well, just about anything you could think of. It was quite expansive and really, really interesting to see. But with all of that, we needed to keep in mind that, once again, this had to be something that is simple, that is clean, fast, and flexible. Something that whatever is built off of this, it could be the building blocks for the future, for whatever it's going to become. Also something very lightweight that if we build something very complex in the back end, we may be stuck with that particular back end. So we put the pieces in place for being able to expand on this one. And this is what you see right here. So everything from the keywords on here that link out and make connections. But that's it for the library side of it. So I'm going to pass it on now.
Thank you very much, Jeremiah. We really appreciate it, and your partnership has been great. So now I'd like to pass it over to our PIs, the COVID Rapid PIs that are joining us today. They're going to do their lightning talks and give us a little perspective on this COVID info comments. First will be Dr. Sarah E.J. Bowman, Associate Research Professor in the Department of Biochemistry at the University of Buffalo, and then Dr. Nora Garza from Laredo College in Texas. Sarah, take it away. Jeremiah, next chart, please.
Okay, great. So can you all hear me?
Yes.
Great. Great. So I'm going to tell you a little bit about some of the work that we're doing in the lab that I direct. I actually run a crystallization center for structural biology at Hauptmann Woodward Medical Research Institute, which is a small nonprofit research institute. And I'm also affiliated with University of Buffalo. Go ahead and next slide. So I'm a structural biologist. We do structural biology, and what that means and what that has to do with the COVID-19 pandemic is that what we're trying to kind of figure out is what all of the different pieces and parts of the SARS-CoV-2 virus really look like. So many of you are probably familiar with this type of figure, which looks large and scary, and it's got these big red spikes on it. Well, that spike is actually a protein, and it's on the surface of the SARS-CoV-2 variant. And it's the protein that interacts with human proteins. And so what we do in structural biology is try to understand what these things actually look like, because it helps us to figure out how to design vaccines and drugs and so on and so forth. So go on to the next slide. Next slide. Can you guys still hear me? Yes. Okay, great. Oops, go back one. Sorry. So structural biology is actually somewhat difficult to do. And in part, this is because the things that we study are incredibly small. And so I like to give people a framework for how big we're actually looking at. So if you think about the width of a single human hair, which is obviously not quite to scale here, but then we ramp down to a single grain of pollen, to one red blood cell, to an aerosol droplet, all the way down to that little pencil mark of a SARS-CoV-2 variant. Now, what we're trying to do in my lab is actually look at what do these things look like in the pieces and parts. And so if the small thing is so small, the pieces and parts are even smaller. And so there's a number of techniques that let us do this. Go ahead and go to the next slide. I'm sorry, they're so don't worry, we're not going to actually talk about the SARS-CoV-2 genome, but we do think about the SARS-CoV-2 genome in terms of what are the different proteins that are encoded by it, because those are the things that we're trying to understand what they look like. So the structural biology community and the whole scientific community, as all of you are probably aware of, have really worked together, very quickly to try to understand a lot of these things. And if you go to the next slide, the reason, again, we're trying to do that is because these are the proteins that actually are treatment targets. So if we can actually develop drugs that will bind to some of these proteins to stop them from acting, we can stop the virus. If we can develop vaccines for that bind the spike protein, and that's actually part of what has been really great in the news lately is some of this work, right? So in structural biology, there's a couple of techniques. Go ahead to the next slide. The one we work on in the lab I work with is X-ray crystallography. And the only thing you need to take out of this is that what we need to do X-ray crystallography is to take our sample and turn it into a crystal. And that turns out to be the bottleneck in the whole technique. And so next slide. So what we do in the crystallization center is we actually facilitate that happening. And the way we do that is in a high throughput way using a 1536 well tray that is essentially the equivalent of what most labs do in a 96 well tray. So it's about 16 times that. And we have really extensive robotics, imaging instruments, and a lot of expertise because we've been running for about 20 years. Next slide. And so we received our NSF Rapid to essentially facilitate researchers from all over the country who are doing SARS-CoV-2 research to try to understand this protein structures, so they could send their samples to us. We could screen them for crystallization conditions, and then they could proceed with their studies. So this has especially been important in the SARS-CoV-2 kind of situation because so many labs are running at really decreased capacity for safety, right? So go ahead, next slide. And so we've really become a major resource we've been a major resource for structural biology, but it's been a really fascinating time to be doing this work with the SARS-CoV-2 proteins. So this is kind of some of the types of images that you get, is to actually view the wells over time, and you can see crystals forming. Sometimes they look like little flowers, like in this one. We actually have multiple imaging techniques that help us to really determine those are crystals. One of the tricky things about this is, though, that we have 1,536 conditions. We have a lot of different imaging over multiple types of images. And so we end up with about 14,000 images for each experiment. And at the moment, right now, people have to look through all of those. So one of the things our rapid funding funded, next slide, was some great work by this fantastic post-bac student, Ethan, who essentially developed a graphical user interface to make it a lot easier to actually view these images, and be able to really interact and work with the data. And he incorporated a really recent machine learning algorithm that identifies these crystal images. So we've submitted a paper describing this software. It's on GitHub, and so available for people. And he recently also got a poster prize for this. So next slide. And we've also had just some tremendous success screening for SARS-CoV-2 samples for users. So these are some of the examples. In the top corner, you see some crystals of one of the proteases, which is a main drug target in complex with a number of different inhibitors. The one with the little heart-shaped picture, that's actually the main protease, the crystal structure that was solved by our collaborators at Oak Ridge National Laboratory. And again, these flower-like crystals appear within, a week. And then we've actually just this week gotten some amazing results on one of the proteins that nobody else has actually had any structural work with. So I didn't have any time. I wasn't able to put pictures up about that, but it's really exciting. So next slide. So it's been really great to be part of the COVID Information Commons as well, because it really facilitates connections. And I think one of the really cool things about this is that, there's a lot of people who are doing a lot of different type of work. So we do structural biology. There's people who are studying all kinds of different things. Already, I can say that I have active SARS collaborations. So this is one of the things that I think Florence will be talking about in a little bit, but essentially you can do searches with keywords to see who else is working on things that are kind of near you. And when I did a search earlier this week on this, I discovered, I actually have 3 active collaborations on SARS-CoV-2 projects right in my little network already. And so it kind of gives you an indication of how well this network is working. And then also, next slide. I also just last week was contacted by somebody who found our work via the COVID Information Commons website. And it's a graduate student in South Korea who's doing research on how data open access during the COVID-19 pandemic kind of compares to other types of things. Her advisor is in the UK. And so we're actually scheduling a time to talk about how all of this stuff works. And so I think that one of the, exciting parts about what's happening right now is all of the connection that is happening, and that is being enabled by kind of what's happening with the COVID Information Commons. So last slide is just my thank yous. So I do want to mention there is a Slack channel for the COVID Information Commons, and we do have a special structural biology channel on that. So if you're interested in any of the things that we're doing, you can come and chat with me there, or check out our website or our Twitter handle. So thank you.
Sarah, that was marvelous. Thank you so much for enunciating the international value. How cool is that? That is just so cool. And I have to say that Sarah, actually, you created the structural biology channel within the Slack channel, didn't you?
Actually, one of my coworkers at HWI did. So
oh, that's what it was.
Who also has a Rapid Award. So.
Yeah, and so that's a really good point, that this is all community driven. So the community is finding this, the international community is finding it. You can create your own sub-channels within the Slack channel to enable collaboration for your focus areas. So we're really trying to make this as community driven as possible. And we're so grateful that the community is getting benefit out of it. So thank you for sharing your story, Sarah, and being part of the community.
Thank you very much. Thanks for having me.
Of course, hang on, we're not done. And so Nora, I'd like to pass it over to you. And as I mentioned, Nora was one of our first stars in the COVID Information Commons community webinars. And her co-PI, Gabriella, is on the line listening in as well. And I'm sure she'll want to thank her. She's been wonderful to work with. So Nora, take it away.
Thank you so much. And at our most recent meeting with our students and our faculty last week, they said, well, thank you to Dr. Garza. And I said, no, thank you to the National Science Foundation for funding us. And a big thank you to the Information Commons, which has made it even more exciting for us and for our students. It's nice to do multi-generational. And so my star is Gabriela Solis Cavazos, who is a former student here at Laredo College, and now she is our undergraduate research coordinator. So our project is using the COVID data that's already there in the City of Laredo website to teach quantitative reasoning skills to undergraduate Hispanic students. Laredo, Texas is right on the border with Mexico. The US came to us. So we've been here a long time, and our college is about 96% Hispanic students. Can you change the slide, please? So we are working with 6 faculty members. Of course, you cannot say it enough. There's a huge stress on all undergraduate students, particularly those pursuing a STEM career. These are hands-on kind of students. They're very high energy. And with the COVID, you know, there were interruptions in their undergraduate research opportunities. They would do bird counts. They would work with simulators. They would work with their faculty, and all of that is gone for now. We're very safe, but we are not meeting face to face. So it's important to retain these students in their classes, even if we have to do it virtually. So we're going to be using that. Can you change the slide, please? When we first got the opportunity to actually write the grant, and I've mentioned it to Florence, I had read Harari and was reading the Heath brothers, and somewhere there they mentioned something about when the impossible happens, think of the possible. And so I like to be on the positive side of everything. I wanted to set up and implement a data analysis research experience to improve quantitative reasoning. Thanks to NSF and our meeting for the HSI project principal investigators, I met Dr. Esther Wilder from New York, and she's been amazing. And she and I in conversation said, you know, when students can really use the terminology, practice it, you know, everyone is impressed. One, they get to practice their vocabulary, and understanding and using quantitative reasoning is really important. So we wanted to create a professional development opportunity for our faculty and in quantitative reasoning. And then we wanted to evaluate the project's impact on our students' quantitative reasoning. So we have faculty from chemistry, from math, from psychology, and from nursing. And they're working with 14 students, and all their projects are related to the COVID. So if you'll change the slide, please. This is what they used to do before. Obviously, they're not out on the river together, but this is Dr. Meng and his students, and soon to be Dr. Celinda Martinez, who I think is at University of Buffalo. Faculty development training. We are reading a book called Math for Life, and we're using it with our students. Oh, and one of our other teachers is teaches statistics. So this is great. Change the slide, please. So you can see there the Laredo, City of Laredo dashboard. And excuse me, these are the posters that students used to prepare before. They're gonna be preparing them again for the projects that they're doing. Change the slide, please. So you have there that we're using the book. We have our faculty. This is a second or third meeting that we have had with the students. And so this past week, 2 of our math instructors, Dr. Carranza and Mr. Carranza, brothers, both math teachers, they talked to them about the y-intercept. And what we were trying to get was, what was the rate of change we wanted for COVID? And that would be to be 0, to flatten the curve. So they're talking about these things, graphing and charting, and it's exciting for our students. we are keeping them motivated. And that was my big worry. I don't know about other universities, but a college, a community college with STEM students, and the situation that we're all living, we have had many students drop out because they have to do other things to take care of their families. Next slide. But Allah, COVID Information Commons community welcomed us. We became a part of them, and our students and our faculty are using everything that's there. And even to write future grants, we are connecting the dots. It is easy to access. If you want to find out any other person that's working on the rapid grants from NSF, you can find it there. The research runs the gamut from research on the actual virus, like the one that you just saw, to the educator and the students' reactions to COVID, what has been happening. So there's all kinds of things that you can access there. A quick perusal will put you in touch with information on informal science learning during these trying times. And of course, Gabriela Solis, who is working hand in hand with our students 10 hours a week. Each student works at least 10 hours a week on their research, and being able to connect to the information commons is very exciting. I know they're all looking forward to tomorrow's lightning talks. Next. And of course, if you have any information on would like any more information, you can contact me at nrgarza@laredo.edu. But you can find more information, of course, on the information commons. Thank you very much.
Thank you, Nora, and thank you, Gabby, who's been in the background and foreground of this whole thing too. So that was wonderful. Both of you were just so inspiring, and this is an example of what the COVID Info Commons is bringing together. We're very fortunate. Next chart, please. So now I'm going to give you a quick run through of some of the search and discovery mechanisms that our colleagues have been talking about. Next chart. So as we mentioned, when we first launched this in July, our minimal viable product, after we got the award in May, we actually launched it with two search mechanisms, the two that you see in the pink boxes on the bottom. So the COVID Research Explorer machine learning generated maps, as well as a customized search by NSF Directorate. And what we try to do, always being community driven, is we listen to what people want. So as you could see, Sarah was using the machine learning generated maps. I'll show you more about that so you know where that came from. And then I've had other PIs who have said that they actually like going and hanging out like in one of the directorates just to see what type of, you see, and Nora's nodding her head, to see what type of research is being funded. And as Nora mentioned, you could look for gaps, you could look for continuity, you could create a constellation out of stars. How could this all work together? And so what we try to do is, provide different tools that are valuable for different people. And one of the things we're planning on is a student hackathon or challenge that we hope to announce in December to do some of the things, Nora, that you said your students are already doing, is have them leverage these different search tools to find opportunities for collaboration, to bring things together, things that are not being done yet, what's missing, and to give us some feedback on the different search mechanisms to see if we should consolidate it or just keep all three of them there, because we've been, building this over time. And we just announced the NSF COVID Awards and PI database, which is really fun. I'll take you through all of these. Next chart, please. So the one that we just launched, as Jeremiah was talking about too, is the awards and PI database. And as you can see, when you go into it, you can get a list of all the awards. You can do it alphabetically by PI, by institution. And then all these things are all the different filters you can use to actually search the awards. You could look by NSF Directorate, by division, by the institution themselves, like Laredo College, University of Buffalo, the state that they're in or territory. We have Puerto Rico in there as well. By region, the 4 big data hub regions, PI name, the program officer at NSF, the program name, reference codes, all sorts of stuff, so that you can actually go in and zero in on what you're looking for. Next chart, please. So when you go into this, you can click on one of those awards and you can see all this information about the award we were talking about. And the really exciting part is you can also click on, you can see how the name of the PI is highlighted. So we'll click on Peter Rose. Next chart, please. And you can see this is his PI profile. And so Nora has one, Sarah has one, And he sent us a fair amount of information. We did the first PI survey. You can see his institution, his email, his ORCID ID, which is a hot link to his ORCID page, so you can see what else he's had going on. The websites he provides that either have results of his research or related to his research. Once they do a PI lightning talk, we add that here as well. So those are also in Sarah's and Nora's. And then we not only have the list of rapid awards, some of them have more than one rapid COVID award, but we have what do they expect their scientific outputs to be, the outputs from their research, collaboration opportunities, and the keywords they use to describe what they do. Because it could be that there are keywords, based on the NSF database, but I always like to ask people, how do you classify it? What is it that you're doing? Because it may be a different way of looking at things. Next chart, please. So that's the PI database, and you could go in and use it as much as you like. The next one, this is a very simple thing, but actually the machine learning maps I'll do first, and I'll do this simple one at the bottom. So these were the two first mechanisms, the machine learning maps for the COVID Research Explorer, and then the NSF Directorate version. So next chart, please. So the machine learning generated maps are based on a tool called Lingo4G Explorer. And when you go into it, you can actually see a topographical map of the NSF Rapid Awards and the topical areas. So this is interesting. And if you look at it and you can see that as an example, protein at the bottom, that looks like a big mountain. So there's a lot going on down there, or social distancing, or surveys, or academic learning. And so you could go in and you can zoom in on this, and you can also click on any of these little dots and actually learn more about that research. And whenever you click on any of the awards, as well as in the next page, you get this information on the right, and you can choose what fields you want: the name of the award, the institution, the PI name, the amount of money they received, their email address, the state they're in, the program officer. There's all sorts of information you can pull up based on what filter you want to use. Next chart, please. Then there's also this view, and this is like the one that Sarah showed, where these are the tree maps versus the topographical map. Next chart, please. And as you go into this, you can actually customize the view you have. So you see here, we can say, I want to color this by this cute little cog up here. You can color by state. I'd like to size it by the amount to see how much money they got. A lot of the rapids are about 200K, so it's pretty consistent. Labeled by institution, and I want to highlight the same color, and let's see what that does for us. Next chart, please. So here I click on one of the institutions in California. So since I said I want to see it by state, I want to see color, state by color, and highlight everything the same color. When I click on a California institution, all the California rapid grants come up. How interesting is that? So if you want to see who else is in your system or in your state, this is a way to do that. Next chart, please. Then you can also look at awards, by institution and in California. So this would be, I asked for the view by institution, and over here in the query, I can say end state California. So it shows me just California. The last page, they colored in the California ones, but there was a whole bunch of them. Here I see just the ones in California. Next chart, please. And so now I can customize the view by PI. So I label it PI. Next chart, please. And then I can actually go in and go for a particular area. So I could say end machine learning or end machine and learning. The joy of Boolean in this case is you get to try it different ways and see what you get. So, the joy of programming for those who've been in it. And so these are all of the ones that are related to machine learning. Next chart, please. And then I could look at, the PIs that are doing the machine learning. And then next chart, please. I can go into the other search mechanism, which would be the NSF Directorate. So here you can click on these icons to find the COVID Rapid Grants by NSF Directorate. The Directorate's Biological Sciences, Computer and Information Science and Engineering, called SCIES often, Education, Human Resources, Engineering, Geosciences, Mathematical and Physical Sciences, Social, Behavioral, and Economic Sciences, and Office of the Director. And they all represent COVID Rapid Grants. So if I click on Office of the Director, next chart, you can see this is actually our COVID Info Commons grant. So it shows you a list of all those in the directorate, and then you can click on this one. Next chart, please. And it gives you similar information as to what we have in the PI and awards database. We've integrated this with the PI information, including the abstract and a lot of it useful information. Next chart, please. So that's a quick overview of the COVID Information Commons, the portal, the community, the PIs that are working in it, the libraries team, and how they enabled us to put the information together the best way that we could to gather the right information from the PIs to make it more useful. And one of the things that we do is really try to follow the FAIR principles, for data in the COVID Information Commons, making it findable, accessible, interoperable, and reusable. And actually, we're going to be presenting at the CoData Go FAIR conference virtually in Paris, only virtually, but November 30th. And we have 9 of our PIs presenting there as well. So findable is what F stands for. So you can find all the NSF Rapid Awards. And we've actually added EAGRs and SBIRs and STTRs for those of you who know some of that nomenclature. And we're looking to add more over the next year that are COVID related. And you can find them, easily in context. It's very accessible. People are using it around the planet, and you can get all these different elements that we talked about. Interoperable from a researcher perspective, we're actually thinking about the next stage of the COVID Info Commons, and Jeremiah and I were talking about it earlier today, but we're thinking of how cool would it be, we haven't figured out how to do this yet, if we could actually crawl the data and help Sarah use her data to find other data in the same type of area, or for Nora and her students to be able to have their data, find data of how is it happening in other states and other HSIs? How does this compare? How cool would that be? So we don't know who can help us code it, but we have a dollar and a dream. And so if anyone knows anyone who can help us with that, Jeremiah are saying we're open, to that. And the data is all reusable. You can download it from the Explorer tool, from the PI award database. There's a CSV file with all the information, and as well you can download in Excel or XML, all sorts of formats from the directorate level information. And we're looking to make that even more useful into the future. Next chart, please. So the next steps, use it. It's your tax dollars at work. You may as well use it. Your kids can use it. Your grandmother can use it. Your friends in Italy, anyone can use it. Join the COVID Info Commons Slack channel if you like. It's on the KIC website. If you go to the covidinfocommons.net, there's a thing at the bottom that says join the Slack channel. You can sign up for the community and the KIC events. There's one tomorrow. There's the bit.ly for it. And help us have researchers collaborate with each other and with students. We're going to be doing the International Fair Convergence Symposium, as I mentioned, at the end of November, 1300 to 1500 UTC, which is now 10 to 12 Eastern. It was 11 to 1 when they sent me the invitation. So luckily we figured that out before November 30th because clocks changed, but UTC doesn't. And you can email us your input or questions at info@covidinfocommons.net. So next chart. So now we have time for Q&A. And we'd be happy to take any of your questions or your answers. We accept both, as I usually say. So if there are any comments, I don't know if there's anything in the chat, Cliff or Diane.
Right. Thanks, Florence. Thank you so much. Well, first of all, thank you for that tremendous presentation, all of you. This is a very exciting tool and just really remarkable, the kinds of synergies that can occur when you bring different sectors of the community together with their various needs and interests. The hub is really exciting. So thank you for this wonderful demonstration of the power of that. And my apologies to everyone. I'm having some technical difficulties, so you'll just get my pretty flower for now and not my pretty face. So with that, would like to invite our attendees to share with us any comments or questions you might have. I think Florence and Jeremiah threw out a challenge there. So if we have anyone in attendance who would like to take them up on that offer, please let us know. And I'll just ask while we're waiting for attendees to weigh in. I'm just wondering, what kind of outreach are you doing to, who are you hoping to draw in? How are you reaching them, other than coming to speak at CNI, of course? How have you been reaching people?
So we try to do it early and often. So we presented at the Academic Data Science Alliance Leadership Summit, which was about a month ago, I think. And so that was a really good discussion. I presented this recently at the UC Santa Barbara Responsible Machine Learning Summit. So people are reaching out, which is wonderful. as I mentioned, we'll be speaking at the International Go Fair Conference later this month. And I think that's a great opportunity to increase the collaboration across the world. I'm hoping that out of that, we have more researchers that start using it on purpose, like the ones that, communicated with Sarah. I hope that we find more data sets, maybe award databases, that we could look at and try to link to. This isn't, even though we're called the big data hubs, we're data innovation hubs. We don't have like big data storage networks and things like that, where we link to everything. So we're more like a collaboration hub. But those are some of the things that we're doing. We're going to be reaching out to all the 990 PIs that are in the new awards database with the PI information to get more of them to give us their survey data so we can enrich it more. We're very lucky that some of the PIs actually did fill out the survey before they could see what we were going to do with the data. But now that they can see, I'm hoping that some more of them say, oh yeah, this is a great idea. We had 170 that, that listened and just gave it to us, which we're really grateful for when we asked for it. So we're reaching out as much as we can. I've spoken on the International COVID research seminar that occurs monthly that's led by the Pittsburgh Supercomputer Center with collaborators from around the world. And we've also just started talking with the COVID-19 HPC Consortium, which works with Exceed and Prace in the US and in the EU. So we're going wide. We want as many people to use this as possible. I like to say this is your tax dollars at work, so use it and help us to help the research move forward, because there's a lot to figure out. When I started this project, NSF actually came to us in March and said, hey, we're thinking of this thing. And I was like, wow, what's that about? We just started talking about it. And I actually at the time did a quick lit review, so to speak. And I found that in 1985, NSF funded, I think it was the second biennial coronavirus workshop in California.
Oh,
I know, right? And I'm like, where's that
Wow.
data? So there's actually a Springer publication with some of the information in 1986. So as a researcher, I look at that and go, man, what's the same? What's different? What did somebody know? And I actually reached out. I tried to find one of the PIs, but they were probably my age back then. But one of them was related to one of the California universities, and they were in touch with him as a retiree. And I asked them to send him a note to see if I could interview him or something. And I haven't heard back yet. But now that we have come so far with this, I'm going to see if maybe he'll talk to us. How cool would that be, that we can share? But some of the things are consistent. You read it says wash your hands, social distance. This was in 1986, so this is not rocket science, as we say, but some of it is, obviously, based on what Sarah was showing us. But there's a lot that we need to learn from the past and predict the future so that we can try not to repeat it the same way, I think. Does that answer your question?
Yes, thank you very much. I think Clifford would like to ask a question now, so I'll hand it over to him.
Yeah, let me just jump in while everybody's thinking about questions. First off, it's wonderful that you've made that connection into the CoData Go Fair thing. I think I've had my eye on that meeting. I think it's going to be very timely, particularly in the kind of context you're putting it in. The question I had, and I asked this a little timidly, not wanting to create any problems, is I noticed on a couple of the slides you had allusions to various NIH programs as well, and clearly while NSF is one of the major funders in this area in the US, NIH certainly is as well. Can you say a little bit about how you're coordinating with or thinking about how what you're doing connects and relates to the funding that NIH is doing in this area?
Absolutely. And as our PI, Jeanette Wing, likes to say, if you think of COVID, you think of NIH, because that's where all the medical and healthcare professionals are. So we really want to partner with them. When we first got this award, it was so much to do in a short period of time. We had to keep our head down and just do the COVID rapid grants, which is what we were told to do. And now we're going more broad across NSF. But one of our next goals is to think about how we can work with NIH and maybe DOE, because even Sarah was talking about the ORNL work that, that she's working with them. And the COVID HPC consortium works with a lot of those different groups. So by collaborating with them, we're hoping we get into their projects. And we actually, the CUIT leader, Carl Ragnoth, and I spent some time last week crawling the NIH website and looking at their search mechanisms. And they don't have an API, but they do have award databases that they download and they update every week. And so now that we're done with getting this all out the door, and our last, October 30th was our last refresh with the new PI database. Now we're going to start looking at how can we bring some of that other data in. And the open public information should be fine, but we do want to open more doors into NIH to start talking about what could we do together, especially if we're thinking about crawling and searching data and metadata, open science data, maybe not HIPAA compliant data. We have to decide what data we're talking about. But I think it would be wonderful to do that more with them. So we do plan on talking more to NIH. NSF has offered to connect us. And I have some contacts there. Actually, tomorrow we have the Computational Approaches for Cancer workshop. I'm on the program committee for at SC virtually. And that's working with NCI. So we do plan on talking more with them because we think we can help these researchers collaborate more and make more progress. So thank you for asking that, Cliff. And any friends and family of NIH that wants to talk to us about this, we're very open and collaborative, as you can tell.
I'm sure you've met Patty Brennan at NLM at some point.
I haven't, but somebody mentioned we should talk to NLM.
You should definitely talk to Patricia Brennan, the director at NLM. I think she'd be thrilled to talk with you about this.
And if you would like to create, like a little letter, if she knows you, if you could send an email, I don't know if you can, I don't want to put you on the spot, but that would
sure.
be cool.
I'd be happy to do that after.
That would be great. Thank you very much. Very thoughtful of you. I'd love to get that. Now we're ready to start talking, past like the heads down, let's get this done approach. So this is perfect timing. Thank you, Cliff.
Sure.
And I don't want to take over Sarah and Nora. I mean, Sarah, you may be working with some NIH researchers already.
Yeah, I mean, so a number of our researchers are NIH funded. And I think that I think maybe earlier this week, I was on a meeting that was indicating that part of why a lot of even the more biomedically relevant funding for the rapids went through NSF was in part because the NSF is really equipped to be able to provide funding that quickly with the rapid award, and that the NIH didn't necessarily have those mechanisms in place. And so I do think that there's going to be more NIH funding kind of available, and so, but it's just a longer process for NIH. So
very interesting. Thank you for sharing that.
Yeah, and I may know some people who might be interested in so I'll touch base with you kind of offline. So
thank you, Sarah. That would be great. Yeah, all friends and family welcome. New friends are making a lot of new friends with this whole effort. So that would be wonderful. Thank you. Any other comments or questions? Jeremiah, did you want to add anything to that? Okay. And he may be having connectivity issues. We're all sharing connectivity, internet, and our computer blew up issues today. So it's really a miracle we got this done. So thank you, team.
There do seem to be goblins in the machine today, for sure.
We're so glad you overcame them and joined us. This has been a wonderful presentation, and I'm really glad that our community had an opportunity to get caught up on this great development.
Well, sure. And they need to thank NSF, and Chaitan Badu was going to try to join us today, but he had another meeting. He's our program officer for this. And I have to thank Martin Halbert for mentioning we should think about presenting, from the hubs, on this. So I have to thank Martin as well.
Yes, Martin is here in the audience.
Thank you, Martin.
Well, I'm not seeing any more questions, and we are at time.
Great, then we're right on time. Thank you so much for having us. And thank you, Nora, Sarah, and Jeremiah for your teamwork. We really appreciate it.
Thank you for having me.
Thank you.
Thanks.
Yes, thank you. Thank you all for coming. I'm sorry, Cliff, go right ahead.
I was just thanking them as well.
Great. I think we'll do what we have been doing, which is go ahead and end the recording at this point, and thank all of our attendees for joining us, and any attendees who want to stay with us here. Please feel free to stay on after we turn off the recording. Just raise your hand. We can unmute you, and you can approach the podium and have a chat with our speakers. And with that, I will be ending the recording and thanking you all and wishing you a lovely evening. Good night.
Thank you.
Good night. Thank you.
Good night. Bye-bye.